Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another DABT topic, that is cellular enzyme biomarkers in blood liver toxicity. So let's directly dive into the question. The question is, what cellular enzyme biomarkers in blood indicate the possibility of liver damage? So we need to identify the biomarkers and we need to also see about the hepatotoxicity. So we have option A, lactate dehydrogenase and liver phosphodiesterase. Option B is mitochondrial pyruvate oxygenase and creatinine kinase. Option C is serum alanine aminotransferase ALT and aspartate aminotransferase AST, and option D is sorbitol dehydrogenase and serum oxygen transferase. The right answer will be in the green text, that is option C, serum alanine aminotransferase ALT and aspartate aminotransferase AST. So let us see what is the explanation and the reference which is given by the American Board of Toxicology. So if you look into these two enzymes, that is alanine aminotransferase and aspartate aminotransferase, are considered two of the standard clinical chemistry tests used in preclinical toxicology studies as indices of a hepatocellular injury. So whenever there is a liver damage, these two clinical chemistry parameters, that is ALT and AST, are the important indices for the identifying if there is any liver injury or not. Next, let us see what other options A, B, and D refers and what are they used for. So from this question, we'll also learn about other type of options, either why they are right or not. So option B, we have lactate dehydrogenase. It is not liver specific, and liver phosphodiesterase is not a biomarker of toxicity. Okay, that is the important thing you have to remember. Even though lactate dehydrogenase levels can be used as a indicator to identify hepatotoxicity, but it is not liver specific, because this can also be used for other types of to identify other types of target organ toxicity. Then comes third one is sorbitol dehydrogenase. Option D can be used to indicate hepatocellular injury. It is not standard test when compared to ALT and AST, which are the standard test. This sorbitol dehydrogenase, it is not the standard test and is therefore not recommended for as a hallmark for hepatocellular injury. Coming to oxygen transferases, serum oxygen transferase, they are not the indicators of liver toxicity, nor are mitochondrial pyruvate oxygenase. Uh, okay, they are not identified, they are not used as a markers for liver toxicity. And coming to creatinine kinase, this creatinine kinase is a biomarker for cardiac toxicity. So whenever there is a toxicity in the heart, the cardiac toxicity, creatine kinase can be used as a biomarker for the cardiac toxicity. Now you might have a little bit of query that serum creatinine as well as blood urea nitrogen can also be used as an indices for identifying okay, those two are used for identify the biomarker for kidney. So whenever there is a kidney as a target organ toxicity is there, at that time serum creatinine, blood urea nitrogen, total bilirubin, many other total protein, like that, many other biomarkers are there to identify kidney toxicity also. So mostly we'll try to make a list of those biomarkers and indicators. So those who are doing researchers know, if they are working on a certain type of target organ toxicity, they should be knowing what type of assay is indicating what type of damage. Okay, so in addition, this American Board of Toxicology has also given the reference Cassett and Dowell, present textbook 9th edition, page number 732 as well as 733. So please go back and read these two particular pages. Let's see what we have in our next slides. So we have taken this particular thing from page number 732 and 733, and the same text is there. So you can read here or you can go back to the textbook and refer. So when the liver is injured, various molecular constituents of the damaged cells. So liver cells are there. Once there is a damage to the liver cell, some of the components from these cells will be released into your blood. So molecular constituents of these damaged cells can be released into the blood. So those constituents we are measuring actually. That is the thing. So these constituents can be quantified in the plasma or serum samples. Okay, either you can measure the plasma sample or serum samples, and they serve as a biomarkers that track the severity of the ongoing liver injury. So what do we have examples? We'll see examples that are used in both clinical and experimental quantification of hepatocellular injury are ALT, that is alanine aminotransferase, and AST, that is aspartate aminotransferase. These enzymes are normally contained selectively within hepatic parenchyma cells, but are released into the plasma when these cells are injured by chemicals or liver diseases. Means these enzymes are already there in the liver cells. Once there is a damage to these liver cells, these enzymes will be released into the bloodstream, and those bloodstreams, either you by measuring in the plasma or serum, we are able to quantify it, or we can able to diagnose it. We can use it as a tool or biomarker. Other enzymes that occur selectively in these hepatic parenchyma cells include mitochondrial glutamate dehydrogenase, ornithine carbamyl transferase (OCT), and various dehydrogenases like lactate dehydrogenase. So these are the differences in these enzymes. Biomarkers of HPC, hepatic parenchymal cell injury, with respect to their concentration, along with sinusoid, organelle association, longevity in the plasma, occurrence in the different species, and selective presence in the liver related to other organs. Okay, so these differences determine specificity as well as sensitivity. Because of these two the nature of the differences, we can determine both specificity as well as sensitivity in detecting and quantifying injury caused by different mechanisms arising from different globular locations or species. So within the species the difference also, and within the liver also there are different lobules are there, no? So but the different lobular location species, we are able to identify the mechanisms. For example, ALT occurs relatively selectively in the liver, whereas AST occurs also in the muscle and other tissues. That's why AST is less selective as biomarker of hepatic parenchymal cell injury. So ALT is more selective for, when compared to ALT, AST also, ALT is much more selectively for liver. Okay. In recent years, there has been interest in liver selective microRNA, example MIR122, high mobility group box 1 HMGB1 protein, and keratin 18 in plasma as hepatic parenchymal cell injury biomarkers. So nowadays new markers apart from ALT, AST, these kind of new, either microRNA or HMGB1 protein or keratin, these are coming up as a new biomarkers, but they have yet to gain utility in clinical settings. Yes, because we have a lot of historical control data based on the previous marker. So whenever a new marker coming into picture, we need to have more number of studies in order to replace the older markers. Okay, so still it is in the pipeline. It's okay. Once we start gathering data, then these new methods can be replaced with the old methods. So in addition to that, you also read table 13.3. Okay, in this they have given classification of drug induced liver injury pattern based on clinical chemistry measurements. Okay, they have given you serum ALT and ALP activity measurements are expressed as multiples of upper limit of normal, then a ratio R of ALT by ALP is calculated. So what is happening is that they have given you some ratios. R is equal to ALT by ALP. So pattern of, so what is the pattern of injury? Whenever the R is greater than or equal to 5, hepatocellular injury is there. Whenever there is R is less than or equal to 2, there is a cholestatic injury is there. Type of damage I'm saying. If the R value is less than 2 and less than 5, it is a mixed. It contains of both hepatocellular and cholestatic. So about these type of R ratio, everything will we have. I have make another video. So in that you can see. For time being, I have kept this one because something related to ALT, AST is here given. That is the reason. In addition, also read this the table 13.4 only. So what they have given is that in this 13.4, characteristic of intrinsic and idiosyncratic hepatotoxicity. So they have given you a list of intrinsic toxicity type A, idiosyncratic toxicity type B they have given. So if you see, with the drugs usually occurs in overdose, distinctive lesion, dose related injury in all individuals, predictable latent period caused by direct effect of agent or its metabolite, reproducible animal models. So all this will comes under intrinsic type of toxicity, type A. Coming to idiosyncratic toxicity, type B, occurs at pharmacological treatment regimens, variable pathology, dose response relationship unclear, susceptible small fraction of people, relationship of exposure to onset of toxicity variable, highly influenced by host contribution, that is example immune response, metabolism, polymorphism, etc., non-reproducible in typical preclinical animal tests. So these are the types of the characteristics of both the intrinsic as well as idiosyncratic hepatotoxicity. So any question comes from this type of also you should be able to answer. So hope from this video you were able to understand about the liver toxicity, important biomarkers, AST and ALT, their significance and R value. So thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel Toxicology on Board. Thank you so much. All the best for your exam.